speaker today.
Thanks, Lauren. So this morning, I'm really excited to introduce Dr. Jess Metcalf, who's an associate professor of ecology, evolutionary biology, and public affairs at Princeton University. Dr. Metcalf is a demographer with broad interests in evolutionary ecology, infectious disease dynamics, and public policy. And her lab focuses on understanding the evolution of immunity and also characterizing complex immunity landscapes in the population for public health purposes. Over the course of the pandemic, she's really been a leader in advocating for how mathematical models can be used to support public health policy, and also thinking about how COVID-19 pandemic might play out over the long term. I'm really excited to have her joining us here today to talk about the risks of vaccine-driven virulence evolution. So please take it away, Dr. Metcalf.
Thank you so much, and thanks so much for inviting me. It's going to be very hard to live up to that introduction, as is often the way. Can you see my screen and my slides? Does that work? Okay.
Yeah.
So really what I'm going to be talking about today is the work of Ian Miller, who's a graduate student with me, and it's entirely his brainchild and his genius. So I want to put that up front. Genius, it's entirely his cleverness. So just to remind us all, and I'm sure nobody needs reminding at this stage, but of course how this works is that SARS-CoV-2 arrives, it binds this ACE2 receptor, enters cells in the upper respiratory tract, starts to replicate. We get worse symptoms if there's replication in the lower lungs. And it's important to note that, of course, immunity also drives many of those symptoms. And if we're lucky, then it's cleared by immunity. There are really three types of variants that matter if we're looking to the future. The things that we care about are increased transmissibility, increased capacity to overcome natural or vaccinal immunity, and increased virulence. And as we've seen for Omicron, even if increased transmissibility comes with a reduction in virulence, it can still be bad because you can still overwhelm hospital systems. But if you're thinking as an evolutionary biologist, there's a clear advantage to increased transmissibility. For any mutant that achieves that is likely to spread. Any mutation that increases capacity to overcome natural or vaccinal immunity increases the numbers of hosts it can infect, especially since, pretty much the sort of end of 2020, maybe when immunity was building up and as vaccines spread through the population. But there's really no direct advantage to virulence alone. Virulence alone, if it's not doing anything else, it's not going to increase the fitness of the pathogen. So the first two are natural things that we expect to emerge and spread. The last has to be happening in conjunction with something else. And of course, I'm sure that I'm telling here people nothing that they don't know, but sometimes it's useful to spell things out very carefully. If for the virulence to spread within populations, it's been hypothesized that what you need is some sort of relationship between virulence and transmission rate. And this might be because if you have higher titers in your lungs, you might cough out more, but you also might be at higher risk of severe outcomes. And this might therefore lead to some sort of relationship between the worse the pathogen was, so the more sick it made us or the more it killed us, the more the pathogen transmitted. But because there's this inherent trade-off in this relationship, so we also expect that if people are dying more frequently, is the classic example that's used, then even though transmission rates might be going up with virulence, we might expect transmission time to go down. And that's simply because people get taken out of the population and so are no longer transmitting if they're very rapidly being killed by the virus, for example. You might immediately think, well, is this really relevant for SARS-CoV-2? Because we know that mortality tends to occur several weeks after infection. The infection period also starts before symptom onset. The majority of individuals experience mild infection, and death is usually several weeks after infection, and is often driven by the immune system. So if such a trade-off does exist for SARS-CoV-2, it's likely to be shaped by something to do with symptoms. So symptom frequency might lead people to isolate more quickly, and thus shorten the infectious period in a sort of slightly less traditional fashion. And we've seen this happen. So we know that the infectious period has changed as people's behavior has changed over the course of the pandemic. So this is not impossible. If you have these two sorts of relationships, you end up with an intermediate fitness optimal. So an increase in virulence, an increase in transmission, but there's less time for transmission to occur in any individual. So that individual is producing more virus per unit time, but it's doing it for less long. So you expect that there's an optimal virulence from the point of view of the virus that's somewhere in the middle of this distribution. So if we want to bring this to bear to thinking about SARS-CoV-2, we need to sort of first answer two questions. Is the evolution of increased transmission possible? And is increased transmission linked to increased virulence? So these two things are things that are necessary conditions for this to even be relevant. And this might seem sort of tremendously naive, but we did start writing this paper before alpha and before delta and before increased transmission was just made manifestly horribly possible in the world. So it's clear that this virus does seem to be able to evolve increased transmission, and it seems to do, you know, R0 seems to increase by a factor of 2 or something every time we obtain a new variant. So that condition is certainly met. The second condition is increased transmission linked to increased virulence? Well, this is much more messy. So it seems that there's certainly, so it was at least estimated that alpha resulted in a 55% increase in the infection fatality rate over the ancestral strain. So it just seemed like there was an increase in virulence there. The last time I looked, and of course the data changes so fast, but last time I looked, there wasn't a huge difference with Delta. It might have been pretty much on the same level as Alpha, possibly a little bit higher. The results were a little muddied. So possible, but certainly not a smoking gun there. And of course, since we published the paper, Omicron emerged. And here, it's very clear that this thing is both more transmissible and less virulent, or is it more transmissible? It's at least, you know, it certainly evades immunity, and there's at least a suggestion that it's not less transmissible, even though it's less virulent. I think that, again, I'm probably entirely out of date already because this is how fast this literature moves, and I apologize, and please do contradict me if people know more recent information on this. But of course, another thing we can imagine is there's no reason to believe that the variants that we're seeing are sitting nicely along these trade-off curves, have gone right up to the edges. Something like Omicron might be sitting at sort of a lower level of transmission. There might be some area to travel before you reach those edges of the spaces at all. So the conclusions from everything we'd seen to this point, and it's incredibly confusing and of course extremely fast moving, but at the time when we were putting this together, it seemed to us that the two things that you could say was that past evolutionary increases in SARS-CoV-2 virulence at least suggests that for future increases are possible, and therefore cause for careful thought. But that certainly the data that was available then, and I think still the data that's available now, are insufficient to conclusively determine if there is a virulence transmission trade-off, its shape, where SARS-CoV-2 virulence and transmission rates fall on this curve, or within the set of strategies that it bounds. So that might be internal as for Omicron. But we still felt that this was interesting to sort of try and bound this problem more greatly. And we felt this in part because, of course, the landscape that the virus is confronting is changing radically, and it's changing radically in part because of the distribution of vaccines. Late 2020, as everyone will never forget, the miraculous news of vaccine development emerged, and they are being rolled out, albeit with inordinate inequity around the globe. Again, none of this, of course, it's a curious thing giving a scientific talk these days because you know that most people know most things that you're saying, but I will say them anyway. So given this change in that landscape of immunity, there is a question, which is what do we expect the future of severity of this infection to be under this changed landscape of immunity, under this change due to the fact that many people will be acquiring their immunity through vaccination instead of through infection. So acquiring immunity the right way, not the wrong way. And this will, of course, hinge fundamentally on how vaccination intersects with the biology of the infection. And the key point here, as I'm sure many of you already see this punchline coming, is that, of course, vaccinal immunity to SARS-CoV-2 could open the door for virulence evolution if vaccination reduces disease to a greater extent than it reduces transmission. So this is a set of a classic result, the classic set of analyses that were first put forward by Sylvain Guindon and colleagues in 2001, where they showed that if they used a sort of simple mathematical model framework to show that if vaccines reduce symptoms, and there is a relationship between symptoms and transmission, you can actually open the door to evolution of greater virulence. And Ian was thinking about this in part because, and this was during the early vaccine trials, it seemed clear that while serious disease was associated with infection in the lower lung, it seemed that some of the vaccines that were being trialed were producing much greater protection in the lower lung and very little protection in the upper respiratory tract. And so there might be this disconnect between transmission and virulence, which is one of those conditions for the evolution of severity under this situation. And the biology underneath why you get this sort of pattern and why SARS-CoV-2 ends up where it does in our respiratory tracts, and why Omicron might have different cellular tropism and end up in different parts is very fascinating, and I think sort of something that we're learning a lot about at enormously fast rates, but is still not entirely worked out. And there's the subsidiary question of how immunity in the upper respiratory tract works and how you can build a sort of transmission blocking immunity on a mucous membrane. It seems like it's a very hard problem still. So we have this potential for the separation between transmission blocking and disease blocking immunity associated with the vaccine. And again, given that the potential, this lays out the possibility that the potential is there for selection for more virulent strains, Ian was interested in pursuing this and sort of figuring out whether one could bound this problem a little bit, particularly given that, of course, we are in a world of global massive inequity in vaccination, which is just a sort of miserable worry. And it's a miserable worry if you believe that vaccination might be selecting for variants that vaccinated people are protected against, but that individuals who have not been vaccinated might be more vulnerable too. And so to tackle this, he built a very simple model that separated out upper respiratory tract and the lower respiratory tract, assuming that some transmission came from both, but that the core part of virulence was associated with infection in the lower respiratory tract. And so fundamentally, you can think about it as two different compartments in the lung, and this was how we started off down this problem. But of course, it's a phenomenological thing, and there's not anything magical about those two parts. Very simple transmission model separating out these two compartments, then what we did was we sort of used a very simple adaptive dynamicsy approach where we looked to see what would happen in terms of the invasion of differently virulent strains, assuming here a relationship between transmission and virulence. And we broadly sort of pinned that into parameter space using what was known about virulence of alpha and delta and what was known about transmission of alpha and delta, so that we looked at variants that started below or above those different points, and with sort of very broad relationships on the curves. And so you can put the results in terms of what happens to severity in terms of protection in the upper respiratory tract. So the proportion of times you might be protecting, if you're vaccinated, you're protecting against transmission, and the lower respiratory tract, so protecting against disease more. As a function of, so different vaccines will sit at different points along these curves. And what we broadly suspect, just given, I gave you the layout, and this is a pretty, you can sort of frame it in words, the logic is pretty clear. If there is greater protection in the lower respiratory tract, that opens the way to selection for increasing optimal virulence in the virus, simply because it can get away with being more virulent, or it is pushed towards being more virulent in vaccinated individuals, because that pushes up transmission, but you end up with much more severe outcomes in unvaccinated individuals. And then upper respiratory tract protection won't do much in terms of that selection range, but might do things in terms of reducing incidence overall, and thus reducing the potential for evolution towards these higher levels of virulence. And this is essentially what the vaccine is doing, is it's removing the cost of virulence to the pathogen, so it can move to a higher optimal virulence. Right. And this is what the patterns look like. So what's shown on the color scheme is the optimal virulence, with the more purpley colors indicating more virulent strains spreading within the population. And the gray areas are areas where there's basically endless selection for virulence, this sort of selection for hypervirulence. It just goes up and up and up. And the white areas are areas on the far right. There are areas where you achieve herd immunity. So you're sort of, pushing down cases far enough so that you're not even in this problem where this evolution can occur. And really, so we, moved all around parameter space and explored it. And this basic schematic comes up again and again and again. And of course, our model was extremely simple and did not include, for example, waning, which could move you around a little bit on this map. And it's hard sort of quite to know what to do with that, except to acknowledge the fact that the spread of these variants seems to be happening very rapidly, even relative to the scale of variance of waning. And so the sort of conclusions that came out of this are nothing sort of shocking. It's that if severity and transmission are linked, and we do not know that this is the case, and it certainly does not seem to be the case for Omicron, if we make pessimistic assumptions around this, just to sort of ground ourselves in the worst case, which does seem to be what happens mostly with this pathogen, possible futures are unbounded selection for increased virulence. You need a very particular set of parameters to get this, so I'm hopeful we avoid this. An intermediate optimal of increased virulence, which seems likely to be a possibility, and then herd immunity that either reduces or blocks the evolutionary potential for the emergence of this virulence. And again, this all hinges on there actually being a relationship between severity and transmission, which across pathogens, across settings, people have come up with some good evidence for it in some settings and some sort of pretty vague evidence for it in others. So there's that to consider. But the big picture, of course, and I know that I'm just sort of preaching to the choir here, is that more circulation globally means more opportunity for variants to emerge, and probably what makes sense more than anything else is trying to drive down numbers of cases by vaccination and NPIs, which will, non-pharmaceutical interventions, which will decrease effective viral population size, diminishing the impact of selection, the genetic diversity of the virus, and thus the likelihood of the emergence of such variants. There may well be, so I was part of this, Julia Gog led this, Julia Gog and Rosego led this group that was asked by the Strategic Advisory Group of experts in the UK as to, whether the UK should be distributing doses, excess doses, and whether you should be targeting them in some way. And of course, it's absolutely impossible to imagine that you would, we just don't know enough to say where you should be sending your excess doses, because there's surveillance is sufficiently poor in particular settings. And although you might think you could imagine ways in which particular combinations of vaccinated and unvaccinated populations, or particular settings where perhaps vulnerable populations or immunosuppressed populations were, we just don't have enough information, I think, at this stage to identify where you should be targeting vaccination in that way. And we do know, one thing we do know is that variants of concern have repeatedly emerged in unvaccinated settings. So distributing vaccines just does not seem like a bad idea. It would be, of course, we'd like to keep track of the future of severity. And we wrote this paper again before Omicron, because everything moves so tremendously, horrifically fast these days. But I think many of the things that we were discussing as we laid this out became manifest when Omicron emerges, which is just how hard it is to estimate infection fatality ratios, because you need populations that are stratified by age and immunological status and other further context status, which of course has been very hard to get to grips with. And of course, there's the other point, which is that we're getting better and better at treating, and we're probably spatially heterogeneous in treating people who are infected. So actually keeping a firm hold on what the infection fatality ratio is quite the challenge. I'm really fascinated by, and I don't know whether this is just a sort of a fascination as an ecologist, but what's going on within hosts seems both incredibly interesting and to potentially have clues as to better understanding some of these things related to virulence. And I do think that there is something to offer from the more evolutionary ecological perspective. Our model was extremely biologically simple and didn't include anything like waning, but also didn't really made this very broad assumption about the relationship between transmission and virulence. And if you think about it, when I was in those first months of SARS-CoV-2, when we were all just reading about coronaviruses all of the time, happy days, which I'm sure we're still all doing. Apparently this introduction suggested that's still where we are. I was fascinated by some of the data that was coming in on what's going on within hosts. And there seemed to be so much that was hinting at the really fascinating patterns of dynamics. There's some lovely work out of Alan Perelson's and a few other labs that placed, for example, the R0 of spread within the lung somewhere between 3 and 8, with a sort of serial interval of around a day. So that's just incredibly fast spread. And you can imagine, just as we have thresholds for herd immunity in human populations, you might have a threshold within the lung where you suddenly ran out of new cells for SARS-CoV-2 to infect. We also know that the virus triggers innate immunity that pulls cells out of circulation because it triggers interferon, which makes them no longer particularly invasible. So those two pieces together, you can sort of start to think about how this spread, this sort of simple concept of an SIR model could describe spread within the lung, and start thinking about how that links to virulence. And there's also some really interesting sort of, immediately quirks emerge, like there seems to be considerable spatial heterogeneity in how cells are infected within infected people's lungs, which would suggest that relatively restricted depletion of target cells, like maybe 60 to 80%, could actually slow or prevent spread within the lung. And so this, I think this gives a sort of window into thinking about how these processes play out. There's also fascinating differences, as I'm sure you all know, in thinking about sex differences in immunity. So the innate immune system seems to respond much more quickly, or at least somewhat more quickly in women than in men. And so if you're thinking in terms of exponential growth, that early response can lead to vastly different outcomes slightly further down the line. Slightly further down the line, of course, is also when other parts of the immune system kick in. And there another metaphor from evolutionary ecology crops up, which is that of predator-prey dynamics, where you can conceptualize readily predators, immune cells, as chasing prey, the viral particle cells, the viral particles, or the virally infected cells. And of course, this metaphor is inaccurate at an incredibly basic level. The survival of predators in this metaphor is not contingent on consuming prey. And affinity maturation makes the predators, the B cells, in some sense, better and better at chasing down the prey and becoming more efficient. But it's likely also that this coupling of predators' immunity and prey could actually lead to a less wasteful response by the host immunity. And some previous work suggests this, sort of looking at how these responses work out. And I think this also, especially in the early days when I was much more steeped in this literature, it seemed that viral titers were the sort of peak viral titer was pretty stable, no matter how fast that early growth was. And if you imagine you have a responding population of predators that is sort of calibrated to the numbers of prey, you might end up with a fairly stable peak viral load, even though that early growth was rather different. Of course, we all love the idea of collective action, which is where simple rule sets at the levels of individuals, so here it would be immune cells, can result in high-level emergent properties. And so much of that early work suggested also that there was massive immune dysfunction going on, that it was hard not to consider that one of the reasons that SARS-CoV-2 was so pathological was that it was disrupting the sort of signaling. And there is also some data coming in on that, but it's very hard to unpick the degree to which specific mechanisms are at play versus a disruption of actually coordinated activity by the immunity. And finally, of course, one of the things that really I found so compelling early on was the fact that nearly identical individuals seem to come up with very different outcomes, so very severe outcomes and very mild infection in others. And of course, this also seemed to happen after viral loads had gone to very low, viral loads had fallen to low levels. And the feedback loops that govern the immune system, in tandem with the impact of sort of friendly fire that the immune system delivers, could drive this. The concept of cytokine storms proved rather tricky to unpick as we moved through the pandemic, because it didn't, for example, map to what we saw in terms of levels of a cytokine like IL-6 in influenza. And it's possible that this is because it's very hard to measure because it's a very dynamic process. And using a quantity like IL-6 at a single point in time might be quite misleading. If you imagine, for example, that you had an inflammatory cytokine that grew in an S shape and perhaps an anti-inflammatory one that grew sort of smoothly, you end up in a situation where you might have two alternative stable states, and sort of slight differences in initial conditions could push you from one to the other. And then you could end up with this, wildly different manifestation of the severity. So I think there's a more and more data to start looking at this. And of course, there's many, many different approaches. There's been some wonderful machine learning work that's sort of unpicking the huge, complicated, horrific landscape of all the immune effectors that people have been measuring. But I think also there might be space for a mechanistic take And there has already been some very, very nice work doing this. And this very nice work also leans a little bit on the comparative context. We've been lucky enough to have 3 different coronaviruses in the last 20 years. This is just showing approximate dates. And of course, the SARS-CoV-2 curve now should be way off the scale. But starting to think about why is it that MERS-CoV, for example, just never seems to turn into a pandemic, even though it's also a spilling over coronavirus. It seems to spill over almost all of the time. I mean, it spills over quite regularly, but never seems to be getting off the ground. And there's many ways in which its fitness might be constrained, but one of them seems to be that it just can't stop being incredibly virulent. And there's lots of potential sort of drivers around that. It seems that the receptor, the equivalent of ACE2 that MERS-CoV, so Middle Eastern Respiratory Syndrome, uses, is actually found, has this gradient in the lungs where it gets to much higher densities lower in the lungs in us. In camels, for no apparent reason, it's the reverse. So it's a very mild infection in camels, and it's addicted to this particular receptor in camels. So it's a very efficient upper respiratory tract infection in camels. That doesn't cause much disease. But in us, if it's using this receptor, then it's condemned to be deep in the lungs, so it's condemned to be very severe, and that might be one of the reasons why it can never get very far, because there's not a lot of time for it to transmit. So there's sort of aspects like this. If there's also some lovely within-host modeling comparative of MERS versus SARS versus SARS-CoV-2. And other features such as the CpG ratio of Middle Eastern Respiratory Syndrome. This is the numbers of Cs followed by Gs, which are at a very low percentage in our own genomes, simply because they tend to be methylated and they're used in particular ways, which makes them a very good label for innate immunity. So innate immunity goes looking for things with low CpG ratios. And MERS, yeah.
Can I ask just a question about this? So how does this mechanistic explanation relate to the much longer serial interval of MERS relative to SARS-CoV-2?
So the lung, I think you've just lovely illustrated how complicated it all is. So it seems that the MERS-CoV seems to grow at much slower rates in the lungs as well. And this might be in part because it cannot invade innate immunity so effectively, because it seems to trigger this, this aspect of innate immunity linked to having higher levels of CpG, but it's all a bit mysterious still. I mean, I don't think we really have that totally unpicked, although actually someone smarter than me probably does have a lot more of it unpicked. But just by chance, SARS-CoV-2 seems to have come out of the animal reservoir that it was in with a much lower ratio of this one sort of honking signal that says to innate immunity, find me and kill me. It just has a much lower ratio, but MERS-CoV-2 is still stuck up at that higher level. So many pieces. So I think, Lauren, to your point, that there's going to be where you are in the lungs. There's going to be how effective you are at getting from cell to cell. There's going to be sort of tropism. There's going to be within host replication. Many different pieces. And I think to get to grips, and this is what I was going to say last, to get to grips with what virulence actually means, I don't think is beyond our reach. I think we're beginning. We should be able to do things like measure the distribution of these receptors throughout our respiratory tracts and those of like possible reservoirs. But you need all the pieces to get a sense of how those things are going together. And so that was my whistle stop tour. I was told 15 minutes. I hope that was true and not 50, in which case we're going to have a very long conversation. Okay. And these are the 2 papers that I've discussed briefly here, and I'd be delighted to chat about this.
Thank you so much. That was really fascinating.